I promise I've worked in a little bit of oncology and pediatrics for relevance to our department here. But today we'll just be talking about in vitro spermatogenesis, a little bit of a review of the rationale, why this could be important, a little bit of what's being done out there, and then what we're doing locally here in our research program. So with that, now to add a little bit of clinical context for everybody doing urology here, about 15% of couples with infertility will suffer with infertility, and a male factor contributes to about half of these cases. Now, the most severe form of male infertility is called azoospermia, as most of us know, where there's no sperm detectable. And this affects roughly about 1% of the population, the general population, or about 15% of infertile couples. So it's actually more prevalent than a person might think. Now, most of us are aware that azoospermia can either be due to a blockage or due to a defect in sperm production. This defect in sperm production is termed non-obstructive azoospermia, or we refer to it as NOA. The etiology for this has only been described in about 15% of cases, while the vast majority are still unexplained. A subset of this population includes cancer survivors who underwent gonadal toxic therapies such as chemo or radiation, and then are rendered infertile. While adults undergoing cancer therapies typically have the chance to access fertility preservation and bank sperm prior to getting chemo, prepubertal and peripubertal pediatric cancer survivors don't have this opportunity to bank sperm since spermatogenesis hasn't initiated for many of them yet. However, in this population, spermatogonal stem cells, which are the essential basis for sperm stem cells, they exist in the testis, and they can actually be biopsied and cryopreserved with a simple biopsy prior to chemo. This is typically done coinciding another general anesthetic, so it's not a lot of additional morbidity or risk to the patient. This is done in some of the major centers in the world, and the tissue is cryopreserved just in case the future technology is developed to be able to regenerate sperm from these stem cells. At the present time, this technology doesn't exist, so it's for future interest, and they've actually done a handful of studies trying to gauge both parents and this pediatric population's interest, and they do find that the overwhelming majority would be interested in pursuing it, even knowing that it might not be possible to use it within their lifetime. Now, treatment for these patients with NOA in general is limited. We can offer a surgical procedure where we open up the testicle in the OR like a coconut, the microtesty. Some of you have probably seen this or perhaps done it. And what we do in this procedure where we open up the testicle is we search through all the small tubules within the testis for basically areas of heterogeneity that might contain sperm. While the most of the testicle does not produce sperm, we're looking for these rare islands that might have focal spermatogenesis that we can pull out, process, and find sperm. Ultimately, this leads to finding rare sperm in about 50% of cases, and this sperm can be used for in vitro fertilization, intracytoplasmic sperm injection, so IVF-ICSI, where sperm is injected into the egg to be fertilized in the lab prior to transferring it into a woman's uterus. Unfortunately, this ultimately only results in a live birth between 10 and 25% of the time. Therefore, there's a significant need for regenerative therapies, as the vast majority of these patients are currently underserved. Now, there are a few potential approaches to sperm production in men with NOA. Obviously, systemic therapies are possible, like giving a drug or another kind of agent, but the blood-testis barrier poses a challenge. And since we're aiming to introduce function, unlike with some of the cancer therapies where we're trying to hit a certain pathway and knock down function, we would likely have to use serial and sequential delivery of numerous molecules in order to progress differentiation of these germ cells. Therefore, harvesting spermatogonal stem cells for culturing and then use in either transplantation into the testis afterwards, xenografting, autologous grafting, or in vitro differentiation in either 2D or 3D culture systems are all potential options that have been tried so far to date. Our interest is to pursue in vitro methods for spermatogenesis, as is the most customizable approach for programming cells and adjusting conditions to potentially rescue some of these phenotypes. Now, from a conceptual basis, to achieve in vitro spermatogenesis, we have to start with a patient-derived testis biopsy. After dissociating it into single cells, we could isolate the spermatogonial stem cells through either magnetic or fluorescent activated cell sorting platforms that are pretty common in the labs. These spermatogonial stem cells can then be expanded, meaning we can grow more and more and more and get them to replicate. And they require unique culture conditions to commit these spermatogonial stem cells to start differentiating towards sperm. Once we get them to commit towards differentiation rather than be in the stem cell population, they would then have to proceed through meiosis 1, meiosis 2, and then followed by spermiogenesis, which is the process where sperm undergo sperm precursor cells undergo major morphological changes to go from a round cell to an elongated tapered head with a tail. And then the DNA has to undergo compaction as well, where it changes from wrapping around histones to wrapping around protamines to make it very tightly coiled. It's driving the numerous stages of germ cell differentiation and maturation that requires a significant amount of research and technological development. And this is where we and other labs are currently focusing a lot of our efforts. So using this approach of in vitro spermatogenesis, it holds several potential benefits over the other approaches for regenerating sperm. Specifically, there's no risk of transplanting cancer cells back into the patient. A xeno-free approach can be taken, and we have recently developed and are validating a xeno-free media to support spermatogonial stem cell expansion in culture. So this just means that we are creating a media or a solution that the cells can survive in and replicate in that doesn't have any animal products. Some animal products can lead to potential contamination with factors that we're unaware of or certain proteins. So if we want to translate it back into the clinical population, ideally we're using a xeno-free media or product. Also, most importantly, perhaps, we can potentially overcome some of the functional deficits in cells through the use of additional growth factors and specific media conditions. Unfortunately, since these are germ cells, using gene editing systems and techniques like CRISPR is not feasible in this type of experimentation, because if we want to translate it to the clinical practice in the future, there's huge ethical problems that go along with this. Obviously, if we're changing the DNA content of the future offspring. However, there are a lot of challenges in successfully being able to achieve in vitro spermatogenesis and reproduce it in humans. Despite having some success in animal models so far to date, where they've been able to achieve in vitro spermatogenesis in mouse models and result in live offspring with IVF-ICSI. Spermatogenesis is otherwise a very complex process, and it requires temporally and spatially coordinated cell signaling, epigenetic changes, transcriptional regulation. So as an overview, the somatic cells within the testis, such as the Sertoli cells, myoid cells, Leydig cells, macrophages, and endothelial cells, they all provide the cytoarchitectural support and signaling milieu to enable germ cells to differentiate through spermatogenesis. Furthermore, the nuances of human spermatogenesis and defective spermatogenesis that we see in NOA require more in-depth understanding. And until recently, we haven't been able to utilize in vitro modeling to assess functional questions. So it was largely just genomics approaches to try to identify a gene. And then if we looked at the tissue level, it was actually very challenging to investigate this because, as I pointed out, there's probably about 20 different cell types, all within one small cross-section of the testis. And if you mash that all together, you end up with an average of gene expression across 20 different cells. So if certain cells are present and certain cells aren't, which we see in NOA, right? There's missing germ cells compared to normal controls. Really, we're just detecting the differences in the types of cells that are present, and rather than the small intricacies in the defective spermatogenesis and the underlying phenotype. So with the advent of single cell sequencing, this has helped provide additional insight. And then animal models has been used extensively in the past, but more recently, trying to utilize some in vitro modeling with human cells, I think is going to move this forward a little bit. Now, before we get too far into the biology and the technologies that have been tested so far to date, it's always important to understand the big picture and some of the assumptions that we're making in taking these approaches, as well as some of the considerations that we really have to keep in mind. So first, when we're attempting to regenerate the sperm production in those with NOA, it's our assumption that these are likely genetic or functionally abnormal germ or somatic cells that may make deriving the sperm more challenging. And it potentially could compromise the health of the sperm if we're successfully able to derive a sperm from the process. Secondly, when we're attempting to regenerate sperm production for cases of acquired NOA, such as these pediatric cancer survivors, we can generally assume that these cells from pretreatment biopsies are normal. However, we may have to overcome immature cell phenotypes, such as the immature somatic cells, which have been well described in prepubertal populations, in order to gain function to help support spermatogenesis to occur in the lab. And then finally, some of the non-negotiables that we need to keep in mind is that while we keep our eye on the goal of regenerating sperm production, we have to think about the implications that go beyond just the act of being able to do this, especially if we want to be able to safely translate this technology back to clinical use. So we obviously can't reintroduce malignant cells back into the host. That would not be great, be obviously dangerous for patients. So that has to be considered. If we're considering autologous grafting or stem cell transplants in particular for that situation, we have to be able to use media and reagents that are xeno-free, like I previously mentioned. We can't use genetic altering techniques in germ cell lines, as this would genetically modify the offspring, like I mentioned with CRISPR. And there are also things that we don't know about using these strategies, such as off-target gene expression profile changes, epigenetic changes to the sperm that are derived through in vitro spermatogenesis, or if there are potential abnormalities that will be translated back to the offspring if we circumvent natural reproductive biology. Is there a reason why failure of sperm production is occurring? And if we can circumvent the natural biology and get these cells to differentiate into sperm, is that going to introduce a risk to the offspring? These are all questions that we'll have to answer upon arriving at successful technology. And while human in vitro spermatogenesis is not complete and reproducible at this time, there are a lot of other centers around the world that have tried many different approaches to date. So with respect to culturing arrangements, some of the others around the world have tried a ton of different platforms in order to support in vitro spermatogenesis. Some of these include 2D culture systems, artificially created 3D scaffold systems, 3D organoids. With respect to culture media conditions, in order to enable basically dynamic cell signaling between different cells and differentiation. They've tried various different base medias, additional gonadotrophins, growth factors to try to recreate some of the cell-to-cell signaling that we identify within in vivo tissue. And this is just an example of many of the different approaches and conditions and reagents that have been used so far to date. Now, there's a lot of different papers published on different approaches. This is one of the more recent ones out of Wake Forest. In this study, as an example, they used adult human testis biopsies. Then what they did is they immortalized the supporting somatic cells. specifically the Sertoli cells and Leydig cells, just meaning that these cells can continue to replicate cell division after cell division after cell division, where typically they end up becoming end differentiated or terminally differentiated. And they don't usually go backwards, except for when we put them in culture, they can regress a little bit so that we can have mitotic activity and get them to divide. They then went on to create organoids with these cells and added the addition of spermatogonial stem cells. Then together, they cultured these organoids for about 23 days. What they found was that the cells organized into spheroids. As you can see in the picture there, the yellow cells are Sertoli cells, red are Leydig cells, and they organize into the periphery, as you might expect, based on in vivo cytoarchitecture. And the blue spermatogonial stem cells organize into the inner layer of the spheroid. They then went on to measure gene expression of some of the classic germ cell markers. And they noted that the postmeiotic cell markers PRM1 and acrosin were significantly upregulated. So this gene here, this is all just fold changes compared to the baseline day zero. Dazzle is just a generic germ cell marker. So there's likely some degree of proliferation and differentiation of these cells. SOSCP3 is a meiotic marker, and then PRM1 and acrosin are these postmeiotic markers that I mentioned. After the gene expression, they went on to confirm these findings with immunofluorescence. And what they found was that PRM1 and acrosin localized within the nuclei, as you kind of expect. So if we're looking at these images here, PRM1 is in green. DAPI is just a generic nuclear stain, just showing where the cell nucleus is. And as you can see, PRM1 is collecting near the edge of the nucleus. Different section here demonstrates PRM1, again, overlapping with nuclei. And then acrosin, again, here, if we look at the far right side, acrosin is in red, DAPI is in blue. And acrosin, we know, contributes to the acrosome, which is on the most leading part of the sperm nucleus. So that was all quite interesting to see. Then for another form of confirmation, they went on to perform digital PCR, which demonstrated that about 0.2% of the cells were positive for PRM1 and acrosin, which are these same postmeiotic cell markers again. So 0.2. ZBTB16 is meiotic, postmeiotic cell markers. So a lot of the cells were in that state. So transitioning a little bit to the research that we're doing in our lab and our approach, what we try to do is build on some of the previous literature and take an approach to build on this previous work, but to work on a few specific questions, all kind of in parallel. First, since we have very little understanding of the pathogenesis of NOA and what's going on a cell-to-cell basis, we want to dive in and understand the cell type specific cellular dysfunction. Secondly, we wanted to functionally assess if NOA-derived spermatogonial stem cells had the potential to be regenerated in the presence of appropriate growth factors and healthy supporting somatic cells. Third, we wanted a better approach to reestablishing the unique 3D architecture. And finally, we appreciate the power of single-cell sequencing in understanding developmental processes and have been working to leverage some of the computational tools to gain insight into the critical drivers of cell differentiation. So to understand cell type specific abnormalities, we first looked at the only single cell sequencing paper evaluating multiple NOA testes. What they found was that Sertoli cells, which largely coordinate and support germ cell differentiation, they were the most abnormal cells, and their transcriptional phenotype was one of immaturity. So we performed single cell sequencing on two subtypes of NOA and found really similar results when we plotted these cells in something called pseudotime, which I'll talk about in a second, when we compare it with normal adults in peripubertal and prepubertal data sets. So this is from the paper that I was mentioning. This is just a plot where the bigger the dot and the more intense the color are two different techniques that indicate cells across the different conditions are most dissimilar compared to normal adult cells. So these are Sertoli cells, big red dots. They're quite different than the others. Not to say that there aren't differences in some of these others, such as peritubular myoid cells, vascular smooth muscle cells, etc. When we looked specifically to reconfirm these results with our Sertoli cells, this is a plot of single cell sequencing. It's called a UMAP. And essentially every little dot on this plot represents a single cell. And within that single cell represents about 1,000 to 2,000 genes of information and their quantitative level of expression. Now, the different colors correspond to different samples in this particular plot. So this top one represents a 7-year-old. The next one is an 11 and 13-year-old. Then there's an adult in the green, which corresponds up here. And then we have two different NOA conditions. One was Sertoli cell only, so no germ cells. And one was late maturation arrest, so it went all the way to spermatids. So what we did is we pulled out these Sertoli cells and we plotted them. As you can see, the prepubertal, peripubertal, NOA, two samples, blue and purple, and normal, all kind of clustered separately, meaning that they were all different enough from each other that they occupied a different space. The two NOA ones were quite similar, so they had some overlap. Now we applied a technique called pseudotime, which basically we have to define what is the end point of differentiation, which we defined as normal adult controls, and what is the most immature state. And of course, we use the youngest sample here, 7-year-olds. And then basically we ask the computer to calculate the rest. So it orders all these cells in between and draws a line. So as you'd expect, go from prepubertal to peripubertal. And then interestingly, we go to these NOA cells, and it looks like there's kind of two branches of differentiation, and then finally up to the normal control. So this just tells us that from a transcriptional perspective, these Sertoli cells from NOA are more similar or closer to an immature state than they are to a final functionally mature state. Interestingly, we also discovered our samples had a large number of interstitial progenitor cells and unique subsets of immune cells, such as T cells and granulocytes, that weren't detectable in normal controls. So this is all our data here. This is another one of those plots like I just mentioned, but instead of the different colors representing different samples, this is a combination of two normal control samples. Each of the different colors now represent a different cell type that we have identified based on their transcriptional profile. So we look for canonical and pathognomonic genes that represent certain cell types, and we can assign these labels. So we have several different somatic cells here, Sertoli, myoid, Leydig, macrophage, and endothelial cells. And then we have this process that looks like a Nike swoosh, largely because it's a developmental process, right? So each cell is undergoing stage-wise specific differentiation from a spermatogonial stem cell all the way through mitosis, meiosis 1, meiosis 2, and then spermiogenesis is all here until we get to sperm. So this is our kind of gold standard normal control. And then we looked at the NOA1 and NOA2. And if you can look at NOA1, this is the one that was basically late maturation arrest or hypospermatogenesis. And here there are Leydig cells and myoid cells, two different populations of Leydig cells. And then we have the other somatic cells here, T cells, macrophages, endothelial cells, and another population of myoid cells, and then Sertoli cells down here. And then basically going from spermatogonial stem cells all the way to spermatids. So the shape of these plots isn't necessarily that important. It's just showing that, again, separation in cell types, but just stating what kind of cell types are present in these populations. And then finally, if we go to the second NOA sample, this is the one that we had a high proportion of interstitial progenitor cells. So these cells had transcriptional profiles of both Leydig cells and myoid cells. And if we look at some of the literature that explored developmental trajectories of pediatric patient testis biopsies, we know that there are progenitor cells that give rise to both Leydig cells and myoid cells. So this is currently what we believe, and we're currently working on validating this cell type. And then we have T cells and granulocytes, again, which we didn't see in the normal condition. So perhaps could there be some immune basis to this? Hard to say at this point, but something that we're looking into. We then went on to further assess the NOA germ cells by comparing them to normal controls when we align them in pseudotime by using something called a trade-seq tool. So basically for that, what this means is we know that we have all these germ cells in normal, and we have these germ cells in NOA. And what we want to answer the question is, what is the difference when we compare apples to apples? So spermatogonial stem cell to spermatogonial stem cell, or round spermatid to round spermatid. And it's quite challenging because when the program creates the thresholds of what it says in a developmental process is a spermatogonial stem cell compared to an early spermatocyte, because it's a continuous process, you arbitrarily have to cut a line based on kind of the statistics in the backing of the program. But what we wanted to do is really make sure that we're trying to compare as homogeneous populations as we could. So this tool basically just allows us to align, if we bin this into tiny little segments in a developmental process, and then we compare bins. It's like having two ladders, and we say, okay, this is where they match, as kind of depicted in this image here. So we're able to match these two continuous processes. So each bin of comparison is hopefully as close to comparing an apple to an apple as possible. So when we did that, what we found was that the NOA-derived germ cells displayed a significant downregulation of base excision repair and nucleotide excision repair pathways, as well as DNA methylation and transcriptional repression signaling, suggesting that there are some deficiencies in the NOA germ cells, in addition to the abnormalities in the somatic cells that I just kind of explained. So the second thing that we wanted to do is determine if there's a regenerative potential for these NOA spermatogonial stem cells, given that perhaps there could be some intrinsic abnormalities. So what we did is we created these testis organoids using somatic cells and germ cells from 4 different conditions. So our negative control, or basically the NOA state, was only using NOA cells, both somatic and germ cells. Second was we use only normal control cells. So this was basically our positive control of taking cells from a patient that had normal spermatogenesis. And then our 2 kind of experimental conditions were using NOA-derived spermatogonial stem cells, combining those with normal somatic cells to see if we can rescue a phenotype with normal supporting somatic cells. And then the opposite, where we took NOA somatic cells to see if they could support normal spermatogonial stem cells, or if they contribute to a deficiency in supporting spermatogenesis from these cells. So far, we've completed this experiment with 1 normal control and 1 NOA patient, and we have a few more replicants currently cooking in the lab. To do this, we expand the somatic cells and the spermatogonial stem cells independently prior to combining them to form the organoids, and subsequently culture them in the presence of spermatogenic promoting growth factors for about 12 days. We found that the normal controls organized appropriately and demonstrated upregulated gene expression for markers of each state of germ cell during spermatogenesis, compared to day 0 when we started the experiments, and they were a suspension of spermatogonial stem cells and somatic cells. So basically similar to that paper from Wake Forest that I mentioned, we did the comparisons of gene expression at the end of our 12-day culturing compared to day 0. And we looked at all these different well-established germ cell markers. So UTF1 and ID4 are spermatogonial markers, SYCP3, MLH3, and to a certain extent CPBP are these meiotic markers, and then TNP1 and PRM2 are post-meiotic markers. When we look at what these organoids actually look like, This is kind of an H&E section of it. And then we stain for different markers. SOX9 is a Sertoli cell marker, so there are plenty of those. ACTA2 are the peritubular myeloid cells. INSL3 are Leydig cells, and SYCP3, as I mentioned, are the meiotic cell markers. Next, we created organoids in each of those 4 conditions I was mentioning. So all normal cells, which are the purple bars on the left side here, all NOA cells, which are the blue bars, then the 2 combinations, which are the green and the orange bars. So again, we performed the quantitative PCR after 12 days of organoid culturing. And we found that upregulation of spermatogenic markers amongst all the states of germ cells were comparable in the normal group and the hybrid organoid group that were containing the normal somatic cells and NOA SSCs. This is the orange group over here. While the pure NOA organoid performed poorly and was comparable to the hybrid organoid containing the NOA somatic cells and normal SSCs. So those are these 2 conditions here. This ultimately suggested, at least for this patient, that the NOA somatic cells were not fit for supporting spermatogenesis, and the NOA-derived spermatogonial stem cells contain some regenerative potential when placed in a better environment. So this orange one was the healthy somatic cells and the NOA germ cells. It looked pretty similar, maybe even a little bit better than our normal control, where the all NOA did not have as much upregulation of the meiotic and post-meiotic markers, similar to when we had NOA somatic cells and normal spermatogonial stem cells. And these are just corresponding organoids from each of these conditions, similar to our previous slide. Our next objective was to create a more biomimetic cytoarchitecture, so something more similar to what we have in our bodies naturally. So we sought to do this by using 3D bioprinting technology in hopes of making an incremental improvement to the 3D organoids and establish the beginning of a more consistent and versatile regenerative platform for performing in vitro spermatogenesis. Our rationale was that first, utilizing supporting somatic cells will provide both the physical cell-to-cell interactions that are necessary for functioning and provide many of the growth factors and cell signaling at a baseline that we wouldn't have to reintroduce and figure out every specific detail as you would if you don't have these cells present. This can be accomplished with 3D organoids as well, but our rationale for using a 3D bioprinting was that it would allow us to create that biomimetic, precise 3D cellular architecture similar to a seminiferous tubule that we see in the testicle. And therefore, this may allow more natural cell-cell interactions and signaling that are necessary to support in vitro spermatogenesis. Also, it would provide a few additional advantages. First, it would allow us to manipulate any cell type prior to combination in a 3D environment if we had to optimize phenotypes, for example, of these Sertoli cells that are immature. If we could create a treatment for these Sertoli cells to make them more functionally similar to a normal cell prior to introducing them, then this gives us that opportunity. Secondly, we could also create hollow tubules to create a unique culturing conditions where we can have a different culture condition or media in the luminal compartment compared to the interstitial compartment. And this is similar to what we see in vivo in the testis. We know those two environments are very different from a cell signaling perspective and media conditions to support each of those cells respectively. And then thirdly, it would potentially allow us to harvest haploid cells, so spermatids or sperm, potentially without destroying this spermatogenic niche that we've created through the 3D printed tube. So what we did is we initiated the experiments by, again, acquiring testis biopsies from patients, making single cell suspensions, and performing differential plating to separate germ cells and somatic cells, then expanding them through several passages. So basically what that means is that once we have the single cell suspension, we put them in a culture dish with certain conditions, and the somatic cells will attach to the plate while the germ cells will float, just given their intrinsic properties of the cell. And then we scoop up the supernatant containing the germ cells. We put them into different conditions so that they will eventually stick down. And the spermatogonial stem cells will propagate with the unique culture conditions. So we then confirmed the cell types that were present after we did this with both qPCR and immunofluorescence using those canonical markers that I mentioned earlier. So as we can see in what we think are SMAC cells, they are showing positive staining for SOX9, MYH11, which is the myeloid marker, and CD34, which is a immune marker. These two markers are what we use for mature Leydig cells. And they didn't show presence here in the 2D culture. And we know that some of these cell types can regress to a more immature phenotype, which is what we believed happened, because when we put them into organoids or 3D prints, we then get re-expression of these two mature markers when they have those cell-to-cell interactions. And then in our germ cell plate, we use multiple different markers of immature germ cells and spermatogonial stem cells, such as CD90, GFR alpha 1, SSEA4, and GPR 125. So those are all spermatogonial stem cell markers. And then STRA8 is a differentiating spermatogonial marker. And then we also tested for the SMAC cells just to make sure we didn't have any contamination. So SOX9 and active2, which we didn't. We next optimized the 3D bioprinting settings using a coaxial needle to achieve hollow tubules. And these range between 300 and 500 micrometer diameters, which are similar to what we see in the human testis seminiferous tubules. So we confirmed this process had very high cell viability after printing, and then compared them to our gold standard 3D organoids. So here, live cells are green, dead are red. And so one of the problems with some 3D bioprinting technologies is that there's a lot of shear stress on the cells, and sometimes it can kill the cells. And if the cells die, it will release all these cytokines and inflammatory things that can impact the rest of the experiment. So fortunately, we eventually got this to work fairly well. And then we compared our 3D printed tubules to the 3D organoids, which have been previously described in the literature. So once we created them, we cultured the organoids and the bioprinted tubules in the same culture conditions for about 12 days. And then we evaluated these germ cell markers for each stage of cell differentiation, like I had previously done with the organoids. And what we found was that the 3D bioprinted tubes outperformed the 3D organoids in nearly all of the germ cell markers. So this first plot is again qPCR gene expression, FGFR3, ID4, or spermatogonial stem cell markers. And we can see the red bars, which are the 3D printed tubes, are much higher on day 12 compared to day 0, suggesting that it can support a spermatogonial stem cell population to proliferate and replicate, or at least have less death over time. Dazzle is a generic germ cell marker, so it's higher. KIT and STRA8 expression are markers for committing spermatogonial stem cells towards differentiation. And then if we look at the meiotic and postmeiotic gene expression, SYCP3, CPBP are the meiotic cell markers, which were a little bit higher or not significantly different here. And then if you look at the postmeiotic markers, TNP1 and PRM2, they're either a little bit higher for the bioprinted or a little bit higher for the organoids. So then we went on to perform H&E staining to evaluate some of the gross cellular organization, which demonstrated that the tubular structure we're anticipating to see, along with positive immunofluorescence for the various somatic cells and SYCP3, which was our meiotic cell marker, these cells were similarly stained between the 3D bioprints and the 3D organoids. So this is kind of what the tube-like structure looks like. We appear to have a basement membrane that's being created. There's a bunch of cells present. There's these little vacuoles or kind of lumen-looking things within the wall. And we think that's from some of the Sertoli cells just kind of reorganizing and secreting fluid to create another little mini tubule within the tubule, which was just an interesting finding. And then if we look at the immunofluorescence, SOX9, these are our Sertoli cells. ACTA2, these are our myoid cells. INSL3, our Leydig cells, and then SYCP3, the meiotic cells. And this just compares to the organoids. This is the image I previously showed to you. So finally, we're moving to the fourth point here. We're evaluating our single cell sequencing data sets of our normal controls using various tools to understand the critical drivers of differentiation to move from between each stage of spermatogenesis. Our thought is that by using these various different approaches, we're hoping that we can find convergent stories by using highly ranked pathways and targets to potentially perform small molecule screens and induce stage-specific germ cell differentiation, which we can then combine with our 3D bioprinting approach to get us a little bit closer to hopefully successfully generating sperm in the lab. Of course, a mountain of safety data will be required when the field finally arrives at developing this technology before it could be implemented into humans as a potential therapy for these couples. But this is what we're currently working on. So in summary, there's a significant need that exists for in vitro spermatogenesis or another technique as a regenerative therapy amongst cancer survivors and men with congenital NOA. Heterogeneity likely contributes to congenital NOA pathogenesis, and is likely why we haven't found, a series of specific genes that accounts for the whole population. Remember, if you recall, about 85% of these patients don't have a known etiology. And with this in mind, it probably means that we need a personalized medicine framework to move this field forward and get a little bit closer to figuring out what's going on and how to fix it. In vitro spermatogenesis has been successfully performed in non-human mammals, such as mice. And there's been a lot of strides that have been taken amongst scientists across the world to try to bridge the gap between our understanding in animal models and apply it to humans. And while spermatogenesis is very similar in different mammals, there are some very critical differences that clearly exist and why we don't have a complete understanding of human spermatogenesis, and certainly not as close as what we have in animal models. I think that single cell sequencing and some of the computational modeling tools will really be critical for moving in vitro spermatogenesis forward and our understanding. And then using 3D organoids, organotypic cultures, 3D bioprinting, they can all serve as very viable platforms to hopefully achieve reproducible in vitro spermatogenesis that could actually be translated more to a clinical setting. And of course, we would definitely need, a lot of safety data and considerations prior to actually applying it to a clinical setting. But the first step is to think about these things in parallel and continue to do the science and development to hopefully arrive at a technology that we can use. So with that, I just want to thank all the team members. Megan Robinson has done just about the vast majority of everything that I presented today in the lab. Arena and Gazelle have been working on the single cell sequencing work in collaboration strongly with Dr. Hack in our department here, Dr. Colin Collins with help with the single cell sequencing, and a collaborator, Dr. Scheibinger in mathematics, who is also helping with the pseudotime computational modeling, Dr. Stephanie Willerth with support with the 3D bioprinting, and Dr. Human Sajri Ardakani from Wake Forest, who published on the 3D organoids and helped get us set up for organoid work. So thank you to all of them and some of the funding that's helped us with this project so far. Happy to answer any questions.